populations are living longer, but unfortunately this has a downside because age is a risk factor for a whole host of human diseases, from neurodegeneration to cancer. This presents serious social and economic concerns worldwide. But what if we could develop medical interventions that target the underlying biological mechanisms of ageing? Could we slow or even prevent ageing altogether? And as a consequence, could we use these interventions to treat multiple diseases of ageing at the same time?
Here, at the Institute of Healthy Aging in University College London and the Max Planck Institute for the Biology of Aging in Cologne, we are trying to understand the causes of aging at the molecular level in order to identify cellular targets for such interventions.
We used the fruit fly Drosophila melanogaster as a model system to study aging. Not because we're interested in how flies age, but because the genetic pathways that influence lifespan have been remarkably well conserved through evolution, from lower invertebrates all the way up to humans. We can therefore manipulate aging in a simple, genetically tractable organism and learn something about human aging. One of the most widely studied cellular pathways in the context of aging is the insulin IGF-1 signalling pathway, a major nutrient sensing pathway and a particular focus of our paper.
The insulin signaling pathway allows an organism to physiologically respond to changes in the nutritional environment. When nutrient levels are high, for example after a meal, insulin or insulin-like peptides are released into the animal's circulation from where they bind to the insulin receptor on the surface of target cells. The insulin signal is transduced through the insulin receptor substrate, Chico, into the cell, initiating a cascade of phosphorylation events through the activation of the lipid kinase, PI3 kinase, which ultimately results in the inactivation of the FOXO transcription factor. Conversely, when nutrient levels are low, insulin is now no longer released into the circulation, so the insulin receptor remains silent. The intracellular phosphorylation cascade is not switched on, and PI3 kinase remains inactive. This relieves FOXO inhibition, so it is now free to bind to and initiate transcription of its target genes. Inactivation of the insulin signaling pathway has important implications for aging, because genetic inhibition of insulin signaling has been shown to increase lifespan in a whole host of model organisms, including Drosophila. And this longevity effect has, until now, been thought to be mediated predominantly through PI3 kinase and FOXO activity.
We now show that in fact the lifespan extension produced by mutation of Drosophila chico also requires inhibition of the RAS-ERK-ETS pathway. We can block the interaction of Chico with either PI3 kinase or RAS and still see lifespan extension, suggesting that inhibition of either signalling nodes is sufficient to extend lifespan.
We also show the D26 transcription factor, anterior open or AOP, which lies beneath RAS and ERK in the pathway, is required for lifespan extension in an insulin signaling mutant. Direct inhibition of RAS or ERK activity also extends lifespan, and we show that this effect is mediated by activation of AOP. Hyperactivation of RAS has an important and prolific role in cancer. Around 30% of all human cancers can be ascribed to its mutation. For this reason, multiple drugs have been developed that inhibit the pathway. We were very excited to find that feeding Drosophila one such compound, called trametinib, which is already in clinical use as a cancer chemotherapy agent, significantly increased their lifespan.
Like insulin signalling, the RAS-ERK-ETS pathway is remarkably well conserved from flies to mammals. It is therefore our hope that trametinib, or another drug like it, could be developed to not only slow aging in humans, but to also increase the health and well-being of older people.